welcome back, everyone. Next, we have NurExone Biologic Inc. It trades on the OTCQB under the symbol NRXBF and on the TSXV under the symbol NRX. It's a pharmaceutical company that's developing a platform for biologically guided exosome-based therapies to be delivered non-invasively to patients who have suffered central nervous system injuries. Please welcome its CEO, Dr. Lior Shaltiel. Welcome, Lior.
Hi, pleasure being here.
All right, the floor is yours. Come back when you're ready for questions.
Great. Again, happy to be here. I would like to present to you today NurExone Biologic. I'm actually calling from Israel. The company is a Canadian-based company, and we are doing cutting-edge technology dealing with regenerative medicine. What does it mean? We are trying to regenerate nerves in the central nervous system, specifically the spinal cord injury. And just recently, yesterday, we published also regeneration of neurons also in the optic nerve. So another field, ophthalmology, but this is our purpose of the company. So when nature ends, NurExone begins. This is exactly that. Move to the disclaimer as we publicly trade company. Take everything what I said in consideration and be aware to the situation that we have publicly traded. Our mission is to develop, as I said, regenerative medicine, and the first product is to target the spinal cord injury, but it's very relevant for other indications of trauma in the CNS, central nervous system. Our first product is XOP10, aiming for a relatively small group of patients. This is why it's orphan drug designation since last year, October. And this drug can regenerate neurons and provide functional recovery for motor and sensoric function of, at the moment, rats, and we want to translate it to humans. Published in 2021, and it's a spin-off from two elite universities in Israel, the Technion, which is the MIT of Israel, with Professor Shulamit Levenberg and Danny Offen from Tel Aviv University. And NurExone has the exclusive license worldwide for the technology. The business is divided to two. We have the exosome therapy, and as a platform, when the first product is the XOP10, and we have the exosome therapy as a platform, the one that we produce our drug, which can be licensed to other or partnership with other companies, dealing with three major things that we have: production 3D of exosomes, loading of exosomes, and what we load exosomes. We are specialized in siRNA therapy, and we use it for our products. NurExone completed 2 years ago in June the RTO, reverse takeover, taking the company that was mining before to become a biological company called NurExone Biologic Inc. And NurExone Biologic Inc. hold 100% of the NurExone Ltd, which is the R&D division that doing all the research and is based in Israel. Several recent activity. We have completed the pre-IND meeting last year, September. We have a detailed guidance from the FDA, what we need to be doing in order to submit successfully the IND, and the plan will be next year to submit it, and what not to do. So very focused, very with the aim to the target and to get it into a clinical stage company. We received in October last year orphan drug designation from the FDA, and we also are working those days to have the same designation in Europe. Just to let you know, from having this designation gives us beneficial, financial beneficial, like a market protection, regardless of IP, tax benefits in the US, approved for pediatric drugs, etc. And also the range of those drugs are much higher than the normal drugs. We are working now to expand our preclinical testing in order to have the IND enabling tests, what the IND needs, or the FDA needs for our getting into human trials. What are exosomes? I'm sorry to say like scientific terms, but it's actually small vesicles, small envelope that the cells secrete. Every cell in the body secrete messages like that, drug delivery system. Why? The next studies I will tell you why. But those are guided missiles. The cells release them in order to go into a damaged tissue. This is this Amazon package with aid for a damaged tissue. When we use it for our benefit, loading them with siRNA to receive a synergistic effect with our therapy. And exosomes become very trendy now because we see a lot of companies working with different different areas like dermatology, ophthalmology, neurology, and different places, and you see the therapeutic effect quite significantly with all those cases. So what is so unique about using exosomes or extracellular vesicles? That's the right terms. They are, first of all, allowed you to have minimal invasive administration. Our proof of concept in rat was done intranasal, through the nose. Through the nose, the exosomes enter to the brain through the BBB, brain-blood barriers, and we find them accumulated in the lesion of the spinal cord. We have a therapeutic effect. It's not an empty drug delivery system. Those are having a lot of goodies that the cells secrete in order to help the damaged tissue. It's allow us to use off-the-shelf technology. Why? Because we don't see any toxology effect. There is very good profile of those those therapy, and we can. It's not like medicine, we are actually can administer the same batch for different people. It's cell-free technology, so there is no cell inside, and help us with the regulation. It's cross the brain-blood barriers, and we are able to externally upload it with our siRNA. So what we do in our platform is that we are taking those exosomes. It's a bit misleading because the exosomes are not empty as you saw before. We are combining in a very easy process of loading with siRNA. You can see like a schematic picture of the siRNA. There's a short sequence linked together with our loading moiety, and this creates exosome therapy nanodrug, which is a drug delivery system with the siRNA that allow us to get into the damaged tissue. And the spinal cord injury is our first clinical indication. First of all, because it does the tech transfer that we made from the university. Second, we wanted to choose this one because it will allow us to receive the orphan drug designation, okay? But it doesn't stop there. We want to get it as a tip of the iceberg to other indications that require regeneration of neurons. What we see actually, and I'm first of all apologize that the video will not work, but you can see the video in the link that you see in the chat. In the video, you will see the full proof of concept that you see after 2 months of treatment or observation, and the treatment was only 2 weeks and the observation was 2 months. You can see the rat walk awkwardly, but walk again after a full transection model. What we see in the middle is what we see in the tissue level. We can see that the cells regenerate and close the gap of the full transection. When you have full transection, you have a gap that doesn't connect the two sides of the nerves. And the third one on the left is the last proof of concept that we made. And actually, it's the third time that we repeat it. And we see the same phenomena again and again. If you see the transection one is damaged, and the nerves is damaged and there is no functionality, and the rats are completely paralyzed in the legs, compared to that everything is compared to the healthy, that everything is intact and there is full functionality. Here in the middle, you see the gap is closed, you see regeneration, and it's translated to a functional recovery. If you compare the XOP10 and his advantage compared to other technology, we can count several. The use of stem cells autologically or allogeneic. We use electrical stimulation and also using monoclonal antibody. In all those cases, when you compare if those drugs are tested in a full transaction model, which is the hardest model, it's a 0-1, or you have regeneration or you don't have regeneration. And it's non-toxic and it's off the shelf, as I said. And we show for the first time using minimal invasive in intranasal administration. So we actually tick all the boxes. Those are the results are based on very intensive science and protection of our IP. We have several layers of our IP. The product is fully protected from the first drug administration, intranasal or minimally invasive of the XOP10 is the granted patent in the US, Russia, Japan, and just recently also in Israel. The second layer is how we reproduce it. We move from 2D to 3D, including production and composition of matter of our exosomes, which composition of matter, by the way, is the highest level of patents. Third is we protect our IP, what we load inside. And the last one is, I think the most important one, is the exosome loading technology. We know how to load the exosomes post-production without any gene modification and no manipulation of the cell. We do it just after the production of the exosomes when they are ready to be loaded. So it's keep this process very simple and very straightforward. As again, the XOP10 is the tip of the iceberg, and I'm very proud now to say that after we press released yesterday the first results of the glaucoma, you can see that we can translate the regeneration capability of the XOP10 from the spinal cord injury to the optic nerves. And this shows exactly the advantages of using this platform for other indication. And I'm calling other company and investors to see the potential of having joint venture partnership licensing of our technology for delivering the peptide or the siRNA of interest of other companies. Moreover, this year in January, we started a first collaboration, which is supported by Eureka for 2 years with a cell therapy that's focused on spinal cord injury with Intelligix. It's a Canadian company that's coming out of the most prestigious spinal cord lab in Canada from Professor Michael Felling. And we are working together to direction chronic to present a better and sustainable solution for chronic patients. What I present before is for acute spinal cord injury, the patients that suffer now. What about the patient that suffered 15 years ago and I don't have a solution for that? We are wanting to find a solution as a combinatorial drug, cell, and exosomes together. And this is what we are working on at the moment in the next 1.5 years. This is the roadmap of our company. The main focus is get the company into a clinical stage. And this is our focus. We are working 100% or more for getting the IND submission next year. And then we expected by the end of the year, also getting into a clinic, first 1/2A clinical trial. Leadership is not only me. I'm the CEO. I maybe show my face and the company everywhere. I'm myself with several degrees in chemical engineering, physiology, pharmacology. Returned to Israel after 8 years in Germany, but I have together with me Mr. Eran Ovadia is the CFO with a track record in biological, large-scale company like Johnson & Johnson. I have together with me the Director of R&D, Dr. Noah Avni, coming from different companies dealing specifically with drug delivery system, from NanoGhost, which is a combined therapy of cells and exosomes, cells and liposomes, sorry. Minovia is drug chondria, etc. So, and she's actually did her PhD with Professor Aaron Chechanover. He's a Nobel Prize honor from Israel at the Technion. Last but not least is Dr. Ina Sarel. She is the head of CMC Quality and Regulation, working together with Proneuron, another spinal cord injury company that tried the clinical stage back in 2014. So she has experience in spinal cord injury and also delivering the technology into a clinical phase. She has a PhD from Boston University in the US. This is our scientific advisor board. We can talk about Professor Shulamit Levenberg, the inventor, together with Danny Offen, and Nachshon Knoller, Professor Nachshon Knoller from the right. He's the neurosurgery professor that came together with Professor Gabriel Zelig from the rehabilitation center. It's work together. The patient needs to go in the end to the rehabilitation center. And it's very important to have a holistic approach, not only the treating the injury, is how to help and get the damage repair with a lot of rehabilitation. We have Professor Michael Belkin. This is also mentioned in our ophthalmology approach. He's ophthalmologist, and we are working together to establish the XOP10 as a therapy for the optic nerve damage. And last is Professor Theodore Dargi. He's a professor from the US, East Coast, neurosurgery, and that he's combined science and being a doctor with a track record of entrepreneur in the US, involved with several companies, and we are working with him to be, the first focal point in the East Coast in the US towards getting our presence in the US even, stronger and larger. These are the directors. We have 3 directors in Israel. I'm included as a director and the CEO. Yoram Drucker is a serious entrepreneur that actually established the company, part of the founders. Dr. Gadi Riesenfeld was the president of Pharmos, also large stage pharmaceutical company from Israel. He lived many years in the US. With us, we have the 2 Canadians directors, Oded Orgil, coming from the capital market, and James Richardson, which is actually a nice story. He was the CEO of our shell company, got in love with our technology, and served us as a director in the board. And with that, I think I will finish. I will skip the share May. So I think it's relatively the same share options, which is relative less than 10%, and then warrants and fully diluted. And I want to thank the audience. And here's the picture, actually, for the first time, ceremony of opening belt at the Toronto Stock Exchange from the holy city of Jerusalem. Thank you so much.
Wonderful. Thank you so much. We do have some questions for you. Marilyn asks, is there a point where nerves are just too damaged to regenerate? Lior, can you hear me?
The reception is not so good, so I cannot hear you well. Yeah, now I can hear you well. Can you repeat, please?
Yes. Marilyn asks, is there a point where nerves are just too damaged to regenerate?
Yeah, so we have a therapeutic window of, I would say, 2 weeks. After that, it's considered to be subchronic and chronic. And in this case, what we have is a scar, developing of a scar, which makes this therapy with using only exosomes very challenging. So using of XOP10 needs to be done very, very quickly.
And Aaron wants you to clarify, does this only work to heal damaged tissues, or can it be used to transport medication to destroy unwanted tissues such as cancer cells?
It's very dependent on the purpose of the target. This is a drug delivery system that can deliver also genes or siRNA peptides that will damage cells. For example, in glioblastoma, brain tumors. It's very dependent what we load inside in order to say what can be the therapy. So in our case, the idea is to inhibit the inhibitor, which if we inhibit the inhibitor, we can create proliferation. We create like what the cancer cells would like to do, to proliferate or regenerate the neurons. But we can also turn it around and say, okay, we want to use those exosomes in order to deliver specifically to cancer cells and kill them. But, this is another innovation.
And are there other companies working on the same concept? And if so, what makes your product better?
So we have several companies working with exosomes. We have the first one that show it in the central nervous system using a unmodified, without gene modification of the cell source. And we are doing it in minimally invasive under a patent. So it's not a systematic approach using IV, or not doing it for dermatology, which is taking it next step of a more challenging path. But we see success, and this is what makes me satisfied that this technology can actually has benefits for many indications. And you can see it also with the ophthalmology approach. If we see a repair of the eyes, that damaged eyes that can actually function as a healthy eyes after 18 days only, I mean, that's remarkable.
Absolutely. Jim wants you to clarify, are you currently in phase 2, and when do you expect your next results to drop?
No, we are at the moment in a preclinical stage. We would like to get into clinical stage next year, and this will be a phase 1-2A. That will be our first trial.
And Alex wants you to review the milestone slide again, specifically talking about what you have down the pipeline the first half of 2024.
Yeah, so as I said, we first things in for the IND, it's dealing with the CMC of the product, characterizing the product. So this we did. Now we need to give the spec of the product, to give the FDA. We are going to produce in X amount of exosomes. What are the conditions? What are the pH? What is the osmolarity? And all the different details of the product. This is what we are doing now. In the second stage is to increase our capacity of preclinical tests, including full tox study in rats, pivotal tox study. And when we have that, we can submit the IND together with the synopsis, how we see our clinical study. So we want to show to the FDA how we to do this clinical study. And this is actually quite complete. We have the synopsis. The idea is to show to the FDA which center we are going to use, and we are going to use several centers in order to complete this phase 1-2A as quickly as possible.
Wonderful. Well, thank you so much for this presentation. Do you have any closing remarks for our viewers today?
I would say stay tuned. We are very active in the social media, LinkedIn, Facebook, if somebody is still there, YouTube, and follow us. And I believe that you will see a lot of results coming in the next coming weeks and months.
Wonderful. Well, thank you so much. And we look forward to having you on again in the future with more updates.
Thank you so much, everybody.
All right. Thank you. Everyone stay with us. We'll be right back.